Today I will brief about enzyme evolution by error-prone PCR and DNA shuffling. Error-prone PCR is a laboratory technique that is used to introduce random mutations into DNA sequences, most commonly for protein engineering and directed molecular evolution experiments. It modifies standard PCR conditions to increase the error rate of DNA polymerase to generate diverse mutant libraries for screening. It is used to create a library of gene variants with random mutations to study protein function, improving enzyme activity, or exploring evolutionary pathways. This technique can be used in protein engineering, enzyme optimization, synthetic biology, and functional genomics. In error-prone PCR, we can use high magnesium chloride concentration, addition of manganese chloride, imbalanced DNTP concentration, or we can use low fidelity DNA polymerase. In error-prone PCR, we can increase magnesium chloride concentration to up to 7 millimolar, and we can add additionally manganese chloride, and we can use imbalanced DNTPs, and we can use low fidelity DNA polymerases so that the mutation rate can be high. And this technique is very important to create random mutations. It creates random mutations in the DNA sequences, so we can screen the function of mutations, or we can combine by DNA shuffling. Error-prone PCR conditions. This is a typical error-prone PCR condition where magnesium chloride is 7 millimolar, and we have added manganese chloride also in the PCR reaction, and primers have been shown, and other conditions are same. This is the cycling, thermocycling parameter. This is also normal PCR cycling parameter. We can check the amplification in agar gel also. It utilizes very normal PCR conditions, cycling condition. This is a typical PCR thermocycling program. 95 degrees centigrade, 4 minutes, 1 cycle. Step 2. In second step 1, 94 degrees centigrade, 1 minute. Step 2, 58 degrees centigrade, 1 minute. Step 3, 72 degrees centigrade, 90 seconds. Stage 3, 72 degrees centigrade for 10 minutes. Construction of mutant library. Mutant library can be constructed after plasmid extraction by alkaline lysis method, vector and error-prone PCR product purification by spin column, digestion by EcoR1, CIP treatment to vector, and then gel extraction, and then ligation, insert to vector ratio 3:1. Construction of mutant library can be continued by transformation, then selection of transformants on LB plates with antibiotic ampicillin as an antibiotic marker. Then we can construct mutant library, and we have constructed a library of 2000 colonies. Screening the mutant library. First, crude enzyme extract preparation. We can prepare crude enzyme extract in a pentaf tube. After inoculating 2 ml LB medium with each colony, then single colony inoculated, then growth was done, then cell pelleting, washing, and desuspending in positive buffer, then adding 1% lysozyme, then 3 freeze-thaw cycles, 5 minutes at icy ethanol bath, and 5 minutes at 50 degrees centigrade. Then crude extract collected was centrifuged, 10,000 rpm, 20 minutes, 4 degrees centigrade. And then enzyme assays and protein concentration was determined. So a typical standard protocol was followed for enzyme assay and protein concentration. Enzyme assays was looked for fructose assay, glucose to fructose conversion. and protein concentration was determined by Bradford methods. Very common techniques to assay protein concentration in any protein samples. Amount of fructose formed was determined by cysteine carbazole sulfuric method. So fructose was looked for enzyme assay. Enzyme purification. Cell was ruptured by sonication. Then centrifugation was done. Then heat treated at 75 degrees centigrade for 1 hour. Then thermal other enzymes, proteins were denatured, and then ammonium sulfate cut was done, 65 degrees centigrade, 65% to 85% saturation. pH study was done for different pHs like pH 5 to 6, 6.3 to 7.5, like this, and enzyme activity was determined at different pH to determine the optimum pH. Then thermostability study was performed. Wild type and mutant enzyme was incubated at 120 degrees centigrade for 0, 5, 10, 15, and 20 minutes. Then the residual activity was determined at 90 degrees centigrade with the possible buffer pH 6.8. And half-life of enzyme was also drawn by plotting a graph. Then DNA sequencing was performed to confirm the mutation. DNA sequencing was done by Shanghai Biotech, and mutation were cited by DNA Man software and compared, and the sequencing graph was compared. These are the kinetic parameters of Zalo's isomerase, like wild type and mutants comparison, optimum temperature, optimum pH, Pmax, Km, Kcat by Km, and below are the half-life of the wild type and mutants. Then two mutations were observed, like asparagine 256 to lysine and histidine 281 to proline. In asparagine 256 to lysine mutation, two hydrogen bonds in wild type, one in main chain and one in side chain, one hydrogen bond in mutant. In histidine, DNA shuffling is done in three stages. First is the randomization with DNase1, where DNase1 enzyme clips randomly the DNA families of genes into smaller fragments, and then after randomization, then this is the randomization protocol where recombinant plasmid is digested with DNase1. This is a typical protocol where the reaction is proceeded at 15 degrees centigrade for 15 minutes. The reaction is stopped adding 6 microliter of 0.5 EDTA and keeping the tip on ice. And in the second stage, fragment reassembly, PCR reaction. This is a PCR reaction without primers. The principle of this PCR reaction is that the smaller fragments that was clipped by DNase1 enzyme act as a primer. So this is a no external primer is added in this PCR reaction. And this is a typical fragment reassembly protocol. And 95 degrees centigrade 90 second, 94 30 second, 58 5 minute, 72 3 minute, and 72 degrees centigrade 7 minute for 35 cycles. Here we have to gel extract the smaller fragments, random digested fragments, and then perform PCR reaction. Then full length amplification PCR reaction with primers. This is a normal PCR mix, normal PCR reaction. PCR program can be like this: 96 per 2 minute, 10 cycles of 94 30 second, 58 30 second, 72 45 second, 14 cycles of 94 30 second, 58 30 second, 72 degrees centigrade, 45 plus 20 second. And this is a electropherogram of a PCR product. Figure 2 is the DNase1 digestion products. Figure 3 is the reassembly products, and figure 4 is the reassembled gene.